Hello, my name is Job Fermi and I am an application specialist at Delmic. I specialize in the development of fast electron microscopy, which is nowadays used a lot in large bioimaging projects where people are interested in looking at larger volumes or large areas. Within this field and within this scope, electron microscopy still suffers from a lot of challenges, like limited acquisition speed, a lack of proper automation, and a lack of robustness, which means that these projects typically have a large overhead and very low throughput. If we're looking how to improve these projects and make sure that the throughput is higher, there are a couple of steps that we need to look at. The first thing is the automation of the microscope itself, because as the microscope goes through the imaging cycle, it needs to make sure that images are always properly in focus and that the stage is in exactly the right position and that artifacts or errors in the system are properly taken care of. These steps are usually repeated for tens or hundreds or even thousands of times throughout the process, so it is important that they do not introduce any overhead. The second part that is necessary is to make sure that the system is robust. And this is to ensure that an operator is no longer needed at all times to mitigate any kind of errors or problems that arise from the microscope. And finally, we need to make sure that the microscope has enough capacity so that the microscope can run for longer periods without needing sample exchange, which is again something that would introduce overhead. So if we combine all these challenges, it becomes obvious that there is a need for a drastic new approach to these kind of projects. At Delmic, we have developed a system that is a solution to all of these challenges. We have ensured a high acquisition speed by using a 64-beam scanning electron microscope, which is further combined with an easy-to-use interface and robust automation to make sure that projects are run unattended and stable for longer periods of time. This system is useful for a variety of applications. It is especially useful for large volume imaging or for large area imaging, which are applicable in many different bioimaging projects. Good examples of these are cell biology, where users will look at cellular architecture, as well as connectomics, where users might look at thousands or hundreds of sections to understand how neurons interact with each other in the brain. If you're interested in more applications or if you're interested in discussing the specifics of the systems, do visit us at delmic.com or get in touch with one of our application specialists.